Hello, everyone, and thank you for coming to my talk on biodisperse MPT, measuring length scale dependent remodeling of hydrogels by HMSCs during migration. So the objective is to use the microbiological technique of biodisperse MPT to measure cellular remodeling of the pericellular region on different length scales in order to better characterize cell migration in synthetic materials that have wound healing applications. We're interested in length scale for several reasons, one of which is that most biological materials are composed of several components which impart length scale dependent properties on them and allow them to have unique cell material interactions, such as cartilage being composed of collagen, aggrecan, and hyaluron. And then we also show that length scale is important in migration. Over the course of the four steps of cell migration, the cell interacts with several different length scales in the material, spanning from 1 nanometer up to several microns. Previous studies have also shown that the pore size of the network is important in cell migration, and as the pore size decreases, you get significant decreases in the cell's ability to migrate. So we use the technique of biodisperse MPT to measure two different length scales simultaneously during cell migration. And we show that the 2 micron length scale is more heavily remodeled than the half micron length scale in cells which are allowed to pull on the network. We then show that the alpha value, or the degree of remodeling of the material, correlates well with cell speed and correlates differently for each of the three length scales, indicating that the cell treats each length scale different during migration. So the technique that we use is biodispersed multiple particle tracking microbiology, and in this technique, video microscopy is used to measure the Brownian motion of fluorescently labeled probe particles embedded in the sample. And then we can use classical tracking algorithms to generate and calculate these different trajectories within the material. And these trajectories have related mean squared displacements, which can be used to relate to different viscoelastic properties of the material. And we've also shown, or others have shown, that the two different sizes of particles can be separated using their radius of gyration, allowing you to calculate two different rheological properties from two different mean squared displacements in the same sample. And the material that we use is an MMP degradable polyethylene glycol hydrogel, whose backbone is this four-arm star polyethylene glycol molecule, which is cross-linked by an MMP degradable peptide sequence, which allows our material to be remodeled by the HMSCs as they secrete those enzymes. And finally, we include this cellular adhesion peptide CRGDS, and also in the precursor solution, we include the photoinitiator LAP, the HMSCs, and the probe particles used for the experiment. We expose this precursor solution to UV light for 3 minutes to give us a 3D encapsulated HMSC within a three-dimensional hydrogel. So some of the results from this work, we've shown that the 2 micron length scale is more heavily remodeled in regions nearer the cell center, as is shown in these two images, than the half micron length scale. And we've shown that over the course of the experiment, we see significantly more remodeling on the 2 micron length scale than on the half micron length scale, especially on days 2 and 3 post encapsulation. We've also shown that the difference in the alpha value, quantified in delta alpha, is significantly different when you inhibit myosin 2, preventing the cells from being able to pull on the material. And this shows that cytoskeletal tension is the source in the increased remodeling on the 2 micron length scale. And finally, we investigated the intersections that occur within the material to identify force centers within the surrounding material. And the half micron intersections occur randomly throughout the field of view in both the untreated and myosin 2 inhibited populations, indicating that these particles are not directed by cellularly applied forces, whereas the 2 micron particles intersect in a small region in the field of view where the cell is applying force, whereas in the myosin 2 inhibited population where no forces are being applied, their intersections also occur randomly within the field of view. We've also shown that the cell speed correlates differently with each particle size on each of the 3 days post encapsulation, indicating that as the material changes on each of the three different length scales, the HMSC is remodeling them differently, and different alpha values are required for the different length scales to achieve a cell speed, a given cell speed. So some conclusions from this work, we've shown that the 2 micron length scale is remodeled by cytoskeletal tension, the half micron length scale is remodeled by cell secreted enzymes, and that the larger length scales require more remodeling for a given cell speed in untreated populations.